Marsilea introduction. Marsilea is a heterosporous aquatic fern having almost 65 species distributed all over the world. The most common species is Marsilea quadrifolia. Habitat. They grow rooted in the mud of marshes and shallow pools. Marsilea vestata and some other species grow in shallow ponds. A single plant of M. vestata may grow in all directions until it covers an area 25 meters or more in diameter. External morphology. The plant body is sporophyte and is differentiated into root, stem, rhizome, and leaves. Roots. One or more adventitious roots are borne at each node on the underside of rhizome. The roots secrete carbonic acid, and because of this secretion the roots could penetrate the shells of pond snails. Stem. The species of Marsilea possess a dichotomously branched rhizome which creeps on or just beneath the surface of the soil. The rhizome is slender, branched, and possesses nodes and internodes. The leaves are borne alternately along the upper side of the rhizome, usually at the nodes. One or more adventitious roots come out from each node on the underside of the rhizome. Leaves. The leaves are compound and borne alternately along the upper side of the rhizome at the nodes. The leaves possess circinate venation typical of most Filicales order of Pteropsida ferns. The leaves of submerged plants possess long flexible petioles and leaf lamina that float on the water surface. The lamina of each leaf is divided into 4 leaflets pinnae arising from the apex of the petiole. At night the pinnae of aerial leaves become folded upwards and exhibits the sleeping movement. Internal morphology. Anatomy. Rhizome. Internally, the rhizome of Marsilea is differentiated into following structures. The outermost layer is single-layered epidermis without any stomata. The cortex is differentiated into outer, middle and inner regions. The outer cortical region consists of compact parenchymatous tissue. This region may be 1 to several celled in thickness. Just beneath the outer cortical region, middle cortical region is present that contains large air spaces, parenchyma. Internal to middle region, the inner cortical region is found. This consists of a sclerotic zone. The cells of this region are thick-walled sclerenchymatous cells, fibers. Beneath this region the cortex again consists of compact parenchymatous tissue acting as storage tissue containing starch. The stele is amphifloic siphonostele. All species possess the siphonosteles externally and internally limited by a single-layered endodermis. Just beneath the endodermis there is a single-layered pericycle. In the center is the pith whose structure depends upon environmental conditions. In submerged species, the rhizomes possess usually parenchymatous pith, whereas the plants growing on mud have more or less sclerenchymatous pith. Petiole. The internal structure of a petiole is as follows. The outermost layer is single-layered epidermis. Just beneath the epidermis there is hypodermal layer. Then there is other cortex constituting of large air spaces, parenchyma, separated from each other by septa. The inner cortex is composed of compact parenchymatous cells. In the central region of the petiole there is stele which is more or less triangular in outline. The stele is surrounded by a single-layered endodermis. Leaflet. The leaflet is bounded by 2 upper and lower epidermal layers. In the case of floating leaves the stomata are confined to the upper surface, but in the case of the plants growing in mud and moist soil, the leaves are aerial, and the stomata are found on both the upper and lower surfaces. Just beneath the upper epidermis there is mesophyll differentiated into palisade and spongy tissues. The vascular bundles are concentric, i.e., xylem elements are surrounded by phloem. Outside the vascular bundle there is single-layered endodermis. Towards lower side there are big air spaces separated by septa. Root. The epidermis is single-layered and composed of thin-walled parenchymatous cells. The cortex of the root is differentiated into 2 regions. The outer region consists of large air spaces separated by septa. The inner zone consists of a few layers of sclerotic cells. Inner to the cortex there is single-layered endodermis. Just beneath the endodermis there is single internal structure of root-layered pericycle. In the center of the stele there is typical dioic and exarch structure with a dimetric plate-like strand of xylem. The phloem bands are found on either side of the xylem plate. Life cycle of Marsilea. Sporangia. Sporocap. Asexual reproduction. Marsilea is heterosporous and spores are produced within the microsporangium and megasporangium. Both the sporangia are produced within special structures known as sporocarps. The sporocarps are borne on short peduncles near the base of the petiole. Each sporocarp possesses many sori which bear microsporangia and megasporangia. The sori are gradate, i.e., the oldest sori are found at their terminal end of the receptacle and youngest at the base. The sporocarps may be oval or bean-shaped. In the earlier stages of its development it is soft and green, but later on it becomes sufficiently hard and brown in color. There are two kinds of spores found in micro and megasporangia, known as microspores and megaspores. Microspores. A microspore is globose in shape with small pyramid-like apex. It possesses a large central nucleus and a large number of small starch grains scattered throughout the cytoplasm. Development of male gametophyte. The germination of microspore takes place quite rapidly, and at ordinary room temperature the microspore develops into a fully mature microgametophyte within 12 to 20 hours. Megaspore. The megaspore is ellipsoidal and possesses a small projection at the anterior end. The anterior protuberant portion contains densely granular cytoplasm and nucleus. The remaining basal protoplast contains a large number of starch grains scattered throughout the cytoplasm. Development of megagametophyte. The development of megagametophyte or female gametophyte is somewhat slower than that of male gametophyte. The megaspore begins to germinate immediately after its formation, and the megagametophyte reaches to its maturity at ordinary room temperature within 14 to 22 hours. Sex organs. Sexual reproduction. Sex organs Antheridium and Archegonium produced on male and female gametophyte respectively. Antheridium, male sex organ, produces many anthrazoids which are multiflagellated. Archegonium, female sex organ, produces single egg. Fertilization. Many anthrazoids are attracted towards the funnel-shaped structure of megagametophyte. The anthrazoids enter gelatinous envelope and reach to the archegonium. Only one antherozoid enters and fuses with the egg to form zygote. Development of the sporophyte or the zygote divides mitotically to form embryo. The development of the embryo proceeds with great rapidity, and only 2 to 4 days old sporophyte possesses the first leaf several millimeters long.